Very, very happy to introduce Dr. Selenica from the University of Kentucky College of Medicine. She will be talking about PAD4 promoting Tau, Citrullination and pathology and Alzheimer's disease. She will be awarded a $125,000 CART grant. Dr. Selenica, the floor is yours.
Thank you very much. Good evening, everyone. I'm very happy to be here. Just let me get to my presentation. So as I was introduced, I am a scientist at the University of Kentucky at Sanders Brown Center on Aging, and I am one of the CART award receivers. And I'm going to discuss for you the role of this protein PAD4 in Tau pathology. But before I start with the presentation, I just wanted again to thank you, the Coins for Alzheimer's Research Trust, for trusting our research and supporting our research. As mentioned by my fellow scientists, that's extremely important for us to move forward to those big findings, and also the bigger NIH funding that we can get to go towards a cure for Alzheimer's disease. And I wanted to start with two perspectives that at least us as a scientist should have and have in mind as we do the research for Alzheimer's disease, and that's the situation of the patients in US today. It is, this is actually the statistics coming out of Alzheimer's Association, and we know that as there are 5.8 million Alzheimer's patients in US today, this number will increase much more with time. In fact, it is, let me do something here for a minute. Take a laser pointer. In fact, it is calculated that by 2050, we'll have as many as 13.8 million patients. And I did mention some of the statistics, which is that, as we all know, age is the risk factor. So as we have 1 in 10 people of 65 years old right now that have Alzheimer's disease, the numbers become 1 in 3 people of 85 and older. We also know that Alzheimer's disease is #1 dementia, but it's also the 5th leading cause of death among the adults at ages 65 and 85. And right now in 2019, the cost of Alzheimer's disease in our society is $290 billion. And that is the cost of healthcare, the long-term care, the hospices. So the Alzheimer's disease, as was described by one of the members of this Rotary, is a devastating disease, and it is a costly disease. It's costly for the patient, for the society. But I wanted to highlight another unspoken, perhaps, part of the Alzheimer's disease, and this is the caregivers. These are the statistics actually show that 83% of people that take care of sick people is actually the family, the friends. And the statistics that came lately is also shown that 46% of those people are actually caregivers that take care of Alzheimer's disease patients. And those are what is called informal care. So the statistics tell us that it's 18.4 billion hours of informal care that these people, this family or friends of Alzheimer's disease patients give to their loved ones. So I do think that the Alzheimer's disease is not only a burden for what I mentioned before, but it's also a burden that is very much on the family, on the friends of these patients. And this graph here just illustrates the percentage of this informal care, this unpaid care that these people give in a different scenarios of the daily living of the patients. And the purple bars here represent the Alzheimer's disease care. And you see that every step of that everyday living, you have a much higher care that is towards those Alzheimer's disease. Something is definitely, okay, so this is the slide that I was talking about, the caregivers and the percentage, an increased percentage of giving care to the Alzheimer's disease patients with the daily activities as is shown here, and the purple bars being the caregiving towards Alzheimer's disease. You guys see that as well? I just want to make sure that I...
Yes, you have it up.
Okay, perfect. I apologize about that. So this is the situation that we have in Alzheimer's disease, and I don't want to spend much time on the pathology of Alzheimer's disease because the speakers before me did an excellent job. Just wanted to highlight again that when it comes to the pathology in the brain of these patients, the two hallmarks is what was described as amyloid beta, that is the core component of this plaque that is in the brain. But another part and hallmark of the pathology is also this protein that is called tau. And tau is a protein that is inside the neuron. And when the pathology becomes very severe, such as this picture is depicting here, you see a neuron that is filled with this tau form that we also call tangle. So I believe many of the audience does know about those pathologies, the tau tangles and the amyloid plaques. And those are the two hallmark pathologies that were actually first described by Alois Alzheimer's, this doctor in Germany, when he described his patient, Auguste Deter. And he started by describing actually her deterioration with Alzheimer's disease. He described her loss of ability to talk, obviously her memory loss, but also depression, which is another severe symptom of this patient. And that was in 1906. Then between 1906 and 1910, after the patient died, they also discovered the plaques that I talked about, and also the existence of these neurofibrillary tangles that are made of tau protein. And then in 1910, this disease was named as an Alzheimer's disease. So just a little bit of history to understand that these hallmarks are really been there from the start. And then I wanted to show this, our community, that the evolution of Alzheimer's disease research. We now it ends here at 2015, but 2020, we have a lot of knowledge, both in scientific findings as well as therapeutic targets and biomarkers that were talked about from previous speakers, and therapies that we all are trying to do to cure Alzheimer's disease. And very few that I wanted to mention that might be of interest for this audience is obviously the ability to image in the brain of Alzheimer's disease and look at this accumulation of amyloid beta or plaques, the ability to look into the brain. And nowadays, and that is actually quite new research, that we're able to track the tau in the brain, as well as look at other, by this major and very complicated technologies, to look into the brain and measure the shrinking of the brain, or to look how well our brain is utilizing glucose, which is the fuel for the brain to work. So this is a very positive picture, I believe, to see how much the research is developed. And then I want to kind of start going back to focusing a little bit on this tau protein, because this is where our research is also focused. What is the tau protein? And what I show here is a very simple sketch of the protein itself that has different regions within it that are important to maintain the stability of the neuron. And what normally, during pathology, what happens with this protein is that it gets many more than it should modifications that are called post-translational modifications of different forms. And those are very well described. All these modifications are known by our community, scientific community, and been described that they contribute in one way or the other towards the pathology that is linked to the tau protein itself. And this graph here pretty much shows the number of different modifications that happen on tau. So those are all the different modifications that happen on tau. Phosphorylation here is the biggest one that we know of, that we have studied, that we have therapies against. And those modifications are actually very important to understand the pathology of tau, because they also become important later on when one has to think about therapies and targeting tau protein. So that's what I wanted to talk in the slide, is about looking at what does these modifications really mean. And those are the emerging breakthroughs in tau recently. And the first one that I mentioned has direct connection to the rest of my talks. I want to spend a few minutes here just to highlight the fact that in 2017, the technique that actually got the Nobel Prize in chemistry was discovering of this little cryo-M title here. So it's a technique that allows you to look at changes of tau in almost atomic level. And they showed that those modifications in the tau protein, so this is the tau protein inside the brain, if you take this tau protein through this cryo-M technique that got the Nobel Prize, you actually can see those modifications on the tau protein. And more important, papers that came through this 2017 to 2020 showed that those modifications on the tau protein actually also determined the different structural diversities of tau. So which means that tau protein is structurally diverse, and those structures can mean something for the disease. But also, before I move forward, I wanted to also mention that knowing those modifications also allows us to use those things as gold standards for biomarkers. We also nowadays have efforts towards vaccines that are going into clinical trials to modify different tau forms. Obviously, the tau imaging in the brain is a big high throughput for the diagnosis of the disease. So it seems that modification of tau protein seems to be very much a key factor into the Alzheimer's disease pathology. So what do I mean by the tau protein structure? I was talking about the modification of tau leading to those different protein structures. So what protein structures do we have in tau? And this is a simple schedule that a figure that I made in terms of trying to explain how tau, this one protein or one single molecule, once on its way to becoming pathological into the severe Alzheimer's disease, goes through different structures. And one that we know in our scientific field, and we know, is this tau oligomer, which is several molecules together, so to speak, that we have shown, our laboratory actually and others have shown that it's quite toxic in the brain. And it starts very early in the disease. Then from this oligomer species, tau can become into a fibril, what is called a fibril. And if you look under the microscopy, this is how the fibrils actually look like. Tau fibrils accumulate more and more and form those tangles that I talked in the beginning of this talk. And here is the picture of the same neuron, a dying neuron that is filled of tangle. And what happens is, as I mentioned in my talk, is that tau is important for stabilizing the neurons. So as these neurons die, then you have this shrinkage of the Alzheimer's brain as you compare to the healthy brain. So again, it seems that modification in tau form and these different forms and structures of tau are important both for maintenance of neuronal function, but also ultimately for the pathology of the disease. The other thing I wanted to mention here before I go into our data that we have found in our laboratory is that we, well, in 1991 actually, Brack and Brack, two scientists, described the spreading of tau pathology. And what we show here is we know now that the spreading of tau pathology in Alzheimer's brain goes through those three different stages, which has a mild. Here in the beginning, you have a mild spreading of tau pathology, then it becomes more severe and goes towards hippocampus, and then the later stages that gets into the cortex and becomes severe Alzheimer's. So we have a description of tau pathology from that mild cognitive impairment that most of you might have heard of to the final and severe Alzheimer's disease. Here, I just wanted to show basically those specific brain regions. So we don't have to go through all of them, but as severe tau pathology becomes, you basically see some of those symptoms that were described and are still described in Alzheimer's disease patients, such as these Broca areas, which is the difficulty of speaking, or the cortical areas, which is those decision-making, as well as hippocampus. And you see actually hippocampus is very important for memory, and that loss in memory is one of the early symptoms that you can see into the patients and measure. So this slide really shows us that as tau pathology increases, that correlates very well with increasing in the cognitive impairment. So tau pathology is, or tau itself as a protein, is then very important to understand and study and have those therapeutic targets towards, as it's so well correlated with the cognitive, the memory loss, and all the symptoms that we see in the patients. So until now, I built this picture that we have very good news coming out of Alzheimer's disease. But we all know, and the reason we are all here is we do know that there is no cure for Alzheimer's disease. So what does one do? As I mentioned, there are many therapies, and of course, those therapies will go forward, and we try to make those better and try to tweak things that perhaps we didn't think through. But the other thing that many laboratories, including mine, is to basically look into unknown pathways, or even pathways that have been understudied or not really fully understood and thrown away. And then one look once again to those and understands that this could be very potential into Alzheimer's disease, because Alzheimer's disease is not a simple disease. It's quite complicated with many, many pathways implicated into the pathology. Actually, the fact that the previous speaker, Dr. Zhao, did about, we now know that TDP-43 is part of Alzheimer's disease, and that was not even believed 2 or 3 years ago even. So this is where our work comes in, really. I wanted to share with you a bit of the discovery of this citrullinated tau by PAD4, and this is what the research that was funded by CART Foundation Trust as well comes into play. So just wanted to introduce those two concepts. So we, in our laboratory, we found, this is a completely new found modification of tau that is called citrullination. And this modification is due to the activity of this enzyme, peptidyl arginine deaminase, or PAD4. I'm going to say PAD4 towards the whole presentation. And what this enzyme does is that it has a client protein, in this case is tau protein, and it removes those groups here on the protein, these amino acids, it removes those and puts some new amino acids into it, which is called citrulline. And CIT stands short for citrullination. So basically you have this change from one group to the other through the activity of this PAD4 enzyme. And the basic scientists have shown in other proteins that when this occurs in a protein, that directly changes the protein structure and behavior. So we are quite interested to understand whether or not that had an effect on tau. Since I mentioned that has never been described on a tau in Alzheimer's disease, so we were very interested to discover that. So I wanted to give the punchline almost in the beginning because I wanted to share with you our findings. Our data basically show for the first time that PAD4 is a target for Alzheimer's disease. PAD4 is increased in Alzheimer's disease, and that makes it very interesting. It becomes a novel and viable target for Alzheimer's disease. Actually, a couple of scientists mentioned that, I think Dr. Lemke mentioned that when certain targets that are known in different diseases and you finally see some correlation with Alzheimer's disease, those are viable druggable targets because already the science behind them is known. And there are certain drugs that are there actually that people use for PAD4 in other diseases or in basic science. So we have a very good target here to look into Alzheimer's disease and see whether or not we can, if PAD4 can become a therapeutic target. Secondly, we found that tau is a client at PAD4. So basically this enzyme uses tau to citrulinate it. And we also have some new data that show, and based on the literature as well, that shows that this new modification, citrullination, determines the tau structure. And just wanted to say that the funding that we get from CART now will help us towards understanding these pathways better, as well as joining the cause towards curing Alzheimer's disease. So ultimately what we want is to develop new drugs that are specific for citrullination, specific for PAD4 to prevent the disease early. I was showing in a previous slide that as tau pathology becomes severe, so does the cognition. So we don't want to prevent it in the end of the disease development. We actually want to prevent the disease quite early. And then secondly, and very importantly, we do believe that citrullination could be used as a biomarker that can help us to detect Alzheimer's disease before the symptoms arise. So what have we done and why do we do these claims? I'm going back to this picture because I introduced for you that we have this tau pathology that is quite mild in the beginning, in those early stages, and quite severe later stages of Alzheimer's disease. So the first thing that we did is exactly that. We actually used some human tissue, post-mortem human tissue from what is called the Bragg stage 1 from Alzheimer's disease patients, and compared it to a human tissue from a stage 6, which is the severe stages of Alzheimer's disease. And we used a technique that simply just stains for these PAD4 levels. So this is as a PAD4 imaging. You really are looking at the levels of this PAD4 in the brain tissue from Alzheimer's disease. And if you look in the early stages, you see barely any staining for this PAD4 protein, while in the stage 6 Alzheimer's disease tissue, we actually see now neurons filled with PAD4. So all it's telling us is that PAD4 levels are increased in Alzheimer's disease brain. And to be rigorous, we actually, and I'm so thankful that the researchers before me actually explained the transgenic animal models that we have in Alzheimer's disease. We turned to 2 different animal models, mouse models of tau. So again, we have these mice that they overexpress human tau, and more importantly, they have certain things that mimic Alzheimer's disease. Mice do not develop Alzheimer's disease. But when you overexpress this human tau, now you have accumulation of tau, you have the mice become forgetful, they don't remember. So we can measure certain things that are within the Alzheimer's disease. So from this mice, we actually, what you're seeing here is a slice from the hippocampus of the mouse. And I did mention perhaps a bit briefly before why hippocampus is because hippocampus is directly linked to the cognitive impairment and that memory, especially the memory impairment. So we looked in naive mice and mice that don't have the human tau versus, as I said, 2 different models of the tau, of the human tau. And again, even in these mice models, we see that although PAD4 levels are somewhat there normally, they're very much more aggressively increased once the tau pathology is there. So we were very interested, knowing the science behind the PAD4 enzyme. We're very interested to show or to see whether or not tau truly was a client of PAD4. So this is simple chemistry in a way. We have an enzyme, which is our PAD4, and a substrate, and that in our case is tau, that we incubate or put those 2 proteins together. And once the enzyme and substrate complex is there, we look at the product. And the product in our case is what we call the citrullinated tau. We did analysis that are called mass spectrometry and found indeed that 13 sites of tau were citrullinated, were incubated with this PAD4. That means that in this system, when you have 2 proteins, tau is citrullinated. But so what, right? What do we show? How to validate that? So what we did, one thing that we actually did is that we collaborated with a biotech company and generated 6 tau antibodies. These antibodies, in theory, in our model that we had, should be specific for citrullinated tau, but not tau and not the enzyme. So to validate that, we simply did this enzymatic reaction again, and then we went on a gel and we ran the PAD4 protein by itself, the tau protein by itself, and then the citrullinated tau. And if I could please highlight the red boxes, basically show that all 6 antibodies here that we generated towards different sites of tau recognize the citrullinated tau, but not each protein by itself. So that was very encouraging for us to at least have antibodies that are specific to these citrullinated tau forms, each of them specific for one different site of citrullinated tau. Now we wanted to go ahead and show that this citrullation of tau actually exists, not only, again, as I said, in a protein level, but in Alzheimer's disease brain. So we went back to that tissue that I just mentioned and looked for one of the forms of citrullinated tau. And again, we show that as in stage 1, we don't see much of citrullination going on, very mildly perhaps. In a stage 6 AD patient, we see neurons filled with citrullinated tau. So that is a new modification that we actually found in Alzheimer's disease. And to be rigorous and to be truthful to our own findings, we went back to those mouse models. Again, this is a non-transgenic animal mouse, a naive mouse, if you will. And then this is a transgenic animal mouse of tau that overexpresses human tau. And again, here we show that the neurons of this tau model are filled with citrullinated tau. So this is encouraging. We knew from the literature that yes, modifications of tau changes the tau structures, and tau structures being the ones that I talked before. So does citrulination changes tau structure, and what does that mean for the pathology? Those are the questions that we had. So we did an experiment here when we aggregated tau. So basically we took a tau protein here and we brought it to this level, the tau fibril. And if you look at this graph, that's exactly what we described. We took PAD4, which we know does not get fibrils by any means, and you see it here, does not aggregate. And we took normal tau and we aggregated it. And there you see, within a couple of hours, tau gets aggregated just as it should, just as I describe it here. We actually see it in this graph here. However, when we took tau and incubated with PAD4, so basically we citrullinated tau, we saw a dramatic, if anything, of aggregation of tau. So tau was unable to create those structures, which one would think that, great, then we don't have formation of the tangles. Well, the picture is a bit more complicated because then we went and looked at other forms of taus. And since our laboratory has an expertise in oligomers, we went and looked at tau oligomers from a technique here. And all we have here is protein that we put on those little dots. And we show basically that when we incubate tau with PAD4, when we citrullinate tau really, then we have increase in those tau oligomers. And as I mentioned from the beginning, we do know that the tau oligomers are quite toxic. They're toxic in cells, they're toxic in the brain. So it seems to us that citrullination inhibits this process, but perhaps enhances this process, which is toxic in itself. So this is kind of our overall hypothesis, as we say in here, is that we do know about this pathway. It's been described and studied very well when tau goes through these different forms to create toxicity, to create neurodegeneration. And I call it tau phosphorylation pathway because it's generally believed that phosphorylation being the major form, but there are many laboratories, including Virginia Lee, for example, that describe acetylation of tau. So there are other modifications that follow this pathway. However, this is our pathway. This is what we actually hypothesize that happens in the brain, which is that we have increase in this PAD4 enzyme, citrullinates tau, it keeps tau into those oligomer forms. And as we know, these forms are even more toxic than the tangles. That in itself is quite toxic for the neuron and causes the degeneration of the brain. So, and I wanted to highlight the fact that if we are right, then we right here have two targets in Alzheimer's disease pathway that we could potentially with time also target. One will be PAD4, and secondly will be the citrullination itself that we can target. So what is our research strategy? Well, we want to determine that PAD4 induces citrullination in a mouse model of tau. And then we ask the question, does citrullination reduces tangle pathology but increases the oligomeric forms? And if it does so, what is the pathological outcomes? And what are the outcomes I do want to investigate? Well, one, we want to look at tau citrullination. As I said, we have 6 antibodies now. We're working towards, and this funding can help us perhaps even to do more antibodies towards tau citrullination. We know 13 sites. We're definitely going to measure the tau phenotype, as I mentioned here, but basically we're going to measure citrullination, but we're also going to measure what happens with phosphorylation. Is there a balance between citrullination versus phosphorylation of tau, and what does that mean for the pathology? Our laboratory is very well known and use behavior measurements, which is we look into the mice and try to see whether or not they are forgetful, their motor skills are there. We also want to measure the inflammatory levels in the brain because PAD4 is involved in inflammation, but also inflammation is a very heavy component in Alzheimer's disease pathology. So with this, I'll stop. I wanted to acknowledge my lab. And this is all of them. Most of the work that is funded by CART Foundation will be done by Dr. Quadri, which is the postdoc in my lab. Daniel Lee is the co-I in this grant, and he has helped along this path, and Will, our collaborators. I wanted to mention that A couple of our preliminary data are generated from Florida Department of Health, which also funded us previously. I was actually located in Tampa, Florida, before moving to UK. And then, of course, the Coins for Alzheimer's Research Trust that thankfully will fund us to basically build more of this story and go to those ultimate goals that we have. So with that, thank you very much.